Genetic mutations include point mutations such as nonsense, missense, and silent mutations. Nonsense mutations is when a premature stop codon appears during mutation, so the rest of the production of the protein is not able to go through. Missense is when you basically replace an amino acid with a different amino acid. This is okay if they are quite similar, such as glutamate to aspartate, and might not have too much damage to whatever the result is. Silent is when there is literally no change, because this is thanks to the degenerative coding of DNA to protein, also known as the wobble theory. And finally, there is frameshift. On the other hand, frameshift includes insertion or deletion. And note that these are when you add or delete an nucleotide in the DNA sequence, and this can cause a shift in the reading of your genetic code, which could be very detrimental because that changes literally every amino acid reading sequence. In terms of inversion, this is when you're looking at the macro scale. The chromosomes can be inverted, it would be cut out and then inverted certain segments. Amplification is when on a large scale you can have certain segments of your chromosomes be duplicated. Translocation rearrangement occurs with non-homologous chromosomes having a recombinant swapping of certain segments of chromosomes. And note that a lot of these can be caused by something called transposons, which are basically jumping genes, as they're known. And there are many different types. There is the IS, there is the complex one, and then there is the composite. There we go. Note that all of them are quite similar in that they have this they will all have a gene called transposase, and these genes will code for an enzyme that will cut the ends of the transposon, and these ends right here are repeating ends, and they are inversed, and they'll cut it out, and then that transposon can then jump out of the genetic code and then insert itself somewhere else. Complex just means that it includes a gene, like a gene A or also a gene B, which could code for something like antibiotics. And composites, transposons, is when you have two IS, and then there's like a central segment of the genetic code, and that can also be cut out as well. So note that transposons are able to kind of cause these types of genetic mutations, such as inversion, translocation, and even deletion.